Hello dear students. In this lecture we will discuss some examples of lethal genes, and this topic comes under Mendelian genetics. So we will take three examples. First of mice, second of plants, and third of animals or humans. So all the genes are not useful to the organisms. There are some genes which develop harmful character leading to the death of the organism, and these genes which are responsible for such lethal actions are called lethal genes. So as the name suggests, lethal genes are not useful for the organism. In fact, they are fatal for the organism, whether it is plant, animal, or any insect. So lethal genes are not of an entirely different nature from the genes we have been studying, but these are the genes whose effect deviates very much from the normal, resulting death. Now, the time at which these genes exert their effect varies. It may be early in the development of the embryo or later, and it also depends on the organ whose abnormality causes death. In human beings, for example, in human beings, defects of kidneys, lungs, and digestive system results in lethal conditions and also results in sometimes death, because these organs do not begin to function until birth. But these genes which causes but some genes which causes any defects in the heart would be lethal in the embryonic stage, because the heart begins to function even in the early stages of embryogeny. So these examples means that depends on where the gene is functioning. If it starts functioning early in the development, so it is fatal. If it starts functioning later, so the person can survive for some years. Most of the lethal genes are recessive and causes death only in homozygous condition. So normal ones, when they are in heterozygous condition and they carry lethal genes, they are not lethal. So the normal ones, when heterozygous carry lethal genes without any lethal effect, and thus they act as carriers of lethal genes. So in heterozygous individuals, such carrier individuals, the recessive alleles which can be fatal are present, but because they are heterozygous, so they are not lethal for the organism, because the dominant allele suppresses the action of the lethal allele. If two individuals or two such heterozygous individuals of these gene mate, then about one fourth of their offspring dies at the birth, and about two-fourth of their offsprings would be heterozygous for this defect, and they will be carriers. So, even though organisms expressing the character do not live to reproduce, these genes are propagated continuously through the heterozygous individuals. So, we will try to understand these with the help of three examples we are taking in this lecture. So first example is of from mice, and we will discuss the example of yellow color. So dominant, this example is called dominant lethal genes in mice. So this was discovered by Q Naught in 1909 when he studied the inheritance of lethal genes in mice. So in domestic mice, the yellow coat color is dominant gene, and say this is by capital Y, and the black coat color is due to recessive allele small y. When two yellow mice are mated, the offspring showed a 2 to 1 ratio of yellow and black mice, and This is a clear-cut deviation from the Mendelian ratio because it should have shown 3 is to 1 ratio if it is following Mendelian ratio. Now, it is shown that the yellow mice are heterozygous for yellow color, and so the mice with homozygous alleles for yellow do not survive, and yellow mice never breeds true. So let us understand example with this with the help of crosses. So you can see here in the first cross the yellow mice, when it is crossed with the black mice, and say the yellow color is yellow mice here in this cross, the male is say heterozygous with capital Y and recessive Y allele, and the black mice is homozygous recessive. So these are the gametes which are formed by a male and female, and so yellow is to black progenies obtained in figure 4.1. Now, but when two yellow mice are crossed, which are heterozygous, so in this cross, instead of 3 is to 1, we get only 2 is to 1 ratio to uh, um, uh, so here you can see that in this cross, here 2 is to 1 is because the homozygous dominant for yellow color dies. It does not survive. It means that this homozygous dominant, when the capital Y has two copies, the mice does not survive and the yellow color is lethal to the mice. The only yellow mice which survive, they are in heterozygous condition with one capital Y and small y. So this is the first example where we are taking the example from mice, and this comes under dominant lethal condition, where in homozygous, when the condition is homozygous dominant with yellow, the mice does not survive. So this changed ratio 2 is to 1 is due to the fact that the individual homozygous for yellow coat color die early. Let us take the second example, which is from plants and which is of maize. So the second example is recessive lethal genes in maize. So first example we have taken is dominant lethal in mice, and this is recessive lethal genes in maize, which is also called albinism in maize. So maize, which is corn, produces white, which is albino individuals which lack chlorophyll, and since they cannot make food in the absence of chlorophyll, these albino plants starve to death. These die in the seedling stage as soon as the reserve food of the seed is exhausted, and L.W. Lindstrom in 1917 showed that green individuals are either homozygous or heterozygous for the character. Now, suppose they are represented by homozygous dominant or heterozygous, where capital W and capital W here represents your green, and small w represents white. So capital W is for green and small w is for white. So capital W is homozygous dominant for green, and this is the next is for heterozygous. So if plants of the genotype capital W, small w, which will be green, they are selfed. Phenotypically, two classes of plants are produced in the ratio 3 green is to 1 white. So here in the cross you can see how these 3 is to 1 ratio is obtained with 3 green is to 1 white. These white plants die after a short period of life because glucose can be administered administered to these white plants, immersing the leaf tips in glucose solution. By such process, the white maize plants, which will die under normal conditions, can be green even to reproduce if we dip leaf tips to glucose solution. So some genetic studies show that the surviving green, which are 1 is to 3, have the genotype which we have just discussed, that these greens are homozygous dominant or the heterozygous dominant greens. So the genotype of white plant which died is homozygous recessive, and this white is regarded as a character recessive to the green character. So the heterozygous green plants are carriers of the recessive gene for whiteness, and these genes are preserved in population and appears again when heterozygous individuals meet. So this is the second example of lethal genes which we have discussed in plants, where the white small w is recessive or white character is lethal to the green gene. Now let us discuss some examples in humans which are lethal. So there are many examples of lethal genes in man, so, but we are discussing some selected examples. So many cases of lethal genes have been reported in man, and some of them are: first example is juvenile amniotic idiocy, when the infants homozygous for this gene appear normal at birth, but they lose eyesight between the ages of 4 and 7. Mental and physical powers deteriorate and they die before or during adolescence. Second example is infantile amniotic idiocy, when the infants homozygous for this recessive gene have an early death due to Tay Sachs disease. Third example is a phenyl ketoniuric imbiquibility, which is idiocy. So individuals with this abnormality have phenylpyruvic acid in their urine, and normal persons do not have this. So the persons are sterile and they have a short span of life, and this abnormality is due to the recessive lethal gene when it is homozygous. So these are the three diseases which you can learn, which are lethal in man. And the fourth example, sickle cell anemia, we have discussed this in class, that this is a autosomal disease, and this is also example of pleiotropy. So red blood cells of few persons assume an abnormal shapes in a medium without oxygen, and this is due to recessive gene when it is homozygous. So such individuals die of anemia at an early stage. This happens in homozygous recessive condition, and it is believed that many prenatal deaths, stillbirths, abortions in man are due to lethal genes. And the fifth example, hemophilia, which is a X-linked recessive lethal, which we have discussed detail in class. So hemophilia is also an example of lethal gene. So the difference between sickle cell anemia and hemophilia is that hemophilia is a sex-linked lethal gene, and it is found more in males than females. Females are generally carriers if they are heterozygous, but males it will express, because this is also a recessive gene or a recessive condition. It is due to a homozygous recessive condition. So you can learn as many examples you want for lethal genes, but in this lecture we have just discussed some selected examples which are in your syllabus. And please read books to understand them more. And thank you, students. Let us meet in next lecture.